Hi everyone, and welcome to our second webinar. I'm Charmaine, the brand manager here at Allozymes, and thanks so much again for joining us today. So just to let everyone know that this webinar will be recorded and uploaded on LinkedIn, so feel free to always check it out for a refresher. So in the next 40 minutes or so, we'll be exploring how AI is transforming we discover and engineer enzymes. So whether you're trying to create a biocatalyst from scratch or really optimize an existing enzyme that is already found in nature for better performance, we'll show you how our integrated approach can help to make that process faster, smarter, and more scalable. So leading today's session is Tiago, our senior computational biologist, who will walk you through how our platform tackles enzyme discovery and engineering from both a digital and experimental perspective. And you also hear from Pradeep, our lead protein engineer, who will share a quick look into how our experimental screening will tie everything together. So as always, we'll wrap up with a Q&A session at the end. So feel free to drop any questions in the chat at any time. And with that, let's kick things off. So Tiago, off to you.
Thank you so much, Charmaine. Welcome everybody, and thank you for joining us today. I'm very glad to be here today presenting to you all. Like Charmaine said, my name is Tiago, and I'm a senior computational biologist here at Allozymes. Just a quick background. So Allozymes was founded 5 years ago by our CEO, Payman, and our CTO, Akbar, as a deep tech startup, and has been rapidly growing ever since. I've joined for around 3.5 years ago, and our headquarters are in Singapore, but we have also recently expanded to Toulouse in France. We are currently a team of over 35 high-skilled and qualified scientists with expertise in all aspects of enzyme evolution, ranging from bioinformatics, biocatalysis, protein and strain engineering, bioprocess, and also downstream processing. Speaking a bit of myself, I obtained my PhD in bioengineering systems from the MIT Portugal collaborative program, and I have since worked and gained experience in diverse business ecosystems, including US corporate, as a computational biologist at Emory's, and also a researcher and PhD in European academic settings, and most recently in this Southeast Asia biotech startup world. I've been developing and applying innovative biotech solutions using computational biology, data science, software engineering, and also machine learning since then. And I'm currently leading Alzheimer's data department. We are helping in the development and improvement of our next generation platform for the rapid evolution of high-performing enzymes powered by our proprietary ultra-high throughput microfluidics system. I'll now let Pradeep introduce himself. Pradeep?
Hi, thanks, Tiago. My name's Pradeep Nair. I'm our lead protein engineer. I've been with Allozymes for about 2.5 years. My own scientific journey, I guess, started studying physical chemistry at UC Berkeley. Then I worked on cancer biology at Genentech. And really my introduction to enzyme engineering was when I joined Novozymes. Novozymes products mostly, the goal is to degrade target chemicals with their enzymes. From there, I moved to the enzymology group in Zymergen. Zymergen also does a lot of enzyme engineering, or did, but their goals were primarily to debottleneck biosynthetic cascades and to improve the efficiency of biocatalysis. So then I joined the Allozymes team in 2023, and I've been working on supporting our data platform and really validating the modeling approaches that Tiago is going to tell us about today. So why don't you lead us off, Tiago?
Thank you so much. Yeah, so today we'll talk and walk you through how we're transforming this enzyme evolution, and with a special emphasis on computational enzyme engineering and artificial intelligence. This is enabling us. This is enabling the rapid, smart design of biocatalysts, and we can start even from the desired reaction without having any real information on the enzyme. We'll be addressing some of Allozymes' internal workflows that enable us to overcome well-known industry challenges by deploying enzyme discovery and computational enzyme engineering pipelines. We'll also showcase Allozymes' capability to design, build, screen, and sequence and experimentally validate this targeted variant library. So it's not only computational biology that we do at Allozymes, it's much more than that. And we're happy to share with you what we do today. As we all know, industrial chemical production is still dominated by dirty synthetic methods, right, involving some toxic reagents, high energy inputs, and poor atom economy. So meanwhile, nature has spent billions of years refining biocatalysts that perform the same reactions and are faster, more precise, and sustainable. Enzymes perform very complex chemistry every day without any major toxic reagents, high heat, or waste. Nonetheless, of course, enzymes are optimized for nature, not industry, and current enzyme development is slow and risky. So whether we are developing some new APIs, some active pharmaceutical ingredients, sustainable materials, or fermentation pathways, the traditional pipeline has several bottlenecks. Some common challenges that we face when assessing new projects are, for example, the severe lack of publicly available research, specifically in groundbreaking approaches, uncurated information on public databases, together with a huge number of potential candidates that are natural enzymes, right? They are impossible to analyze individually. And at Allozymes, we go way beyond just finding an enzyme. It's not trivial to find enzymes that perfectly execute industrial processes, as I've mentioned, and when we find a suitable enzyme, there's still the need to engineer it to improve its performance. Our platform tackles this enzyme development from two fronts mainly. First, by discovering candidate enzymes for reactions with no known catalysts, and then by optimizing these same candidates with enzyme engineering. So we support our experimental work with in silico approaches to initiate, accelerate, and also refine what we do in the wet lab. So this makes our workflows a bit more systematic, data-driven, and also very adaptable to different needs. Starting with just a substrate product pair, for example, we can generate and prioritize enzyme candidates for testing using structural predictions, the database screening, molecular modeling, and machine learning. So where we go from? When we encounter a case where, for example, no information other than the target reaction is known, we deploy our first phase, which is our discovery workflow, to find a suitable starting backbone. This is done mainly by mining information on very large pool of enzyme information and processing, filtering, and ranking potential candidates. How do we do this? So our discovery workflow deploys a mixture of approaches to retrieve rich information of the target reaction and also from potential enzyme candidates. From literature research to metagenomic and curated database mining, sequence similarity search, sequence alignments, we create clusters of enzyme candidates predicted to carry at least similar chemistry, even if they are uncharacterized in the literature, for example. We then retrieve and restore all of this detailed information for further analysis. So what we do with this information, once we have our initial candidate pool that can have a very large number of potential candidates, we process each candidate's information leveraging state-of-the-art deep learning models. This enables us to accurately predict the three-dimensional structure and also predict the target substrate's most probable binding site. With this, we retrieve for each candidate the most likely active site coordinates. With this information, and at this point, we can also narrow down the list of candidates based on mechanistic constraints and rank the remaining candidates according to their potential to catalyze the target reaction. So with all this retrieved information on both the candidate enzyme and the target reaction compound, we can perform molecular docking simulations and calculate the target substrate's binding affinity towards each candidate individually. With this, we create a ranked list of calculated catalytic activity. This process, for example, enables us to go from potentially thousands of naturally occurring enzymes with multiple origins to a refined shortlist of around 40 to 50 structurally characterized enzymes. At this point, we are ready for experimental validation and initial catalytic characterization. And so Pradeep is now going to guide us on how this is performed in-house.
Great, thanks, Tiago. So once we have the results of our discovery pipeline, we basically paired that list of potentially thousands of homologs with potential to be active against our target substrate. We've paired that list down to something on the order of tens of homologs. We then synthesize those genes, clone them into the desired host for the project. It might be E. coli, it might be Pichia, whatever is needed. And then we assess the activity, the enzymatic activity of each of these homologs in the needed application conditions. So here's an example where a customer had a target compound that they wanted to react with an enzyme to produce a desired product. So our discovery pipeline identified 40 homologs. We then developed an assay to measure the product that was formed. In this case, it was HPLC, so high performance liquid chromatography. This is a way to use analytical chemistry tools to directly detect the product. And we expressed each of these genes in the desired host and then assessed whether there was any activity, whether the product was formed. And we were able to rank these 40 homologs in terms of how much product was formed in a number of different conditions that were meant to predict the activity in the final application needed by the customer. So from this discovery pipeline, we are able to get to a good starting point. And I want to emphasize it's a starting point usually. Nature has evolved enzymes that are used by organisms to survive, but it's often not the same conditions as needed by the customer for their final application. So usually once we have a starting point, we'll want to fine tune that starting point enzyme to find something that's really tailor-made for the final application. So we're going to walk through a case study now where we use the second part of our pathway or our pipeline, which is enzyme engineering. So in this case, we had a desired product. This is styrene oxide shown here. Styrene oxide can be synthesized from styrene and peroxybenzoic acid. So it's a dirty chemistry to make, but there is commercial value to this molecule. And we wanted to see whether we could synthesize this enzymatically. So we were able to find a starting enzyme that could synthesize styrene oxide from styrene, but it also can synthesize 50 other products from more than 15 different substrates. So this class of enzymes was unspecific peroxygenases. They get their name because they are promiscuous. And so we wanted to see whether we could engineer specificity into this unspecific peroxygenase. So with this target in mind, we wanted to see what mutations could be introduced to this wild type produced by nature that would create an enzyme that would specifically generate styrene oxide from styrene, but not as much of these side products shown here. And Tiago can tell us how we went about engineering this enzyme.
Sure. So as Pradeep was saying, one part is selecting a suitable backbone, a candidate enzyme that is able to catalyze our reaction. The next part is engineering to our specific needs, and that's a huge part, mainly because to mutate just 10 positions in an enzyme, each of which with the available 20 natural amino acids, we are looking at around 10 trillion potential combinations, right? So this is definitely the major bottlenecks that exists at this point. It's simply the immeasurable scale of mutational space. And that's not even a particularly large region in the enzymes, since enzymes usually have hundreds of residues. In order to screen variants experimentally so we need to narrow this focus, right? We need to narrow the focus and select the substitutions on the enzyme that will have the highest impact in improving our activity, our target activity. So this is where our in silico enzyme engineering workflow comes in. Having found like I said, a suitable natural occurring enzyme, we need to evolve it towards our target performance. So to solve this, our in silico enzyme engineering is deployed to find the optimal set of mutations to evolve our target enzyme to the desired activity. And so how do we find this optimal set of amino acid substitutions, right, that will improve our target enzyme activity? Our computational enzyme engineering workflow is a smart, AI-driven, but also structure-informed pipeline. It uses a custom mixture of approaches, combining state-of-the-art deep learning models, evolutionary conservation information, and also structure-based models. So all of these to highlight the mutable hotspots in our target enzyme sequence. Depending on the proposed objective, of course, we integrate multiple tools to point us to the handful of residues that are truly worth mutating. So this approach generates what we call a virtual scanning library, which we then can screen and process to rank and select high potential hotspots in our target enzyme. For that, we use, like I said, state-of-the-art deep learning models of different architectures. So, for example, protein large language models can be trained that are trained on massive protein sequence databases. With this, we can assess each individual mutation evolutionary likelihood or each mutation's weight on the catalytic performance towards a specific substrate, for example. So, I'm talking about different models that have different targets. If there's a need, we can also fine-tune models with a small performance data set to generate more targeted predictions. But not only AI is used in this workflow, a crucial step of our in silico enzyme engineering workflow is the rational approach that we take. So leveraging on the enzyme's predicted 3D structure when there's no characterized structure, and the identified active site, we can rationally observe and select the location of each hotspot. We can look at the active site and try to measure its diameter, try to check if our target substrate fits in there. And with this window, looking into the actual structure of our target enzyme, we can also perform more in-depth molecular docking simulations, for example, and also use, say, a subset of different variants to rank combined positions, or compare, for example, binding affinity of different other substrates of interest to, for example, in this case, try to decrease promiscuity to a specific, or try to increase specificity to a specific target. So to summarize, the deployment of this workflow and the identification of highly important sequence hotspots helps us rationally design combinatorial libraries with high potential for activity improvement. So this enables us to focus our design efforts on a very small subset of positions. It's typically around 5 positions per library, and prioritize that are prioritized based on functional likelihood, structural compatibility, and evolutionary plausibility, making these library designs far more efficient and directed. These libraries are then synthesized by Pradeep using synthetic biology.
Great. I should say it's not just me doing all of the synthesis, but we do have a team that will get the pieces of DNA that we need to introduce the target mutations, and then we introduce these mutations into whatever was our target backbone enzyme. In this case, it's a UPO enzyme that we were working with. And then we clone these gene variants into our desired host. So that might be E. coli, it might be Pichia, it might be Cerevisiae. Each host has different advantages and disadvantages. In this case, we were working with Pichia. It's often chosen because it secretes large quantities of protein, so it's easier to isolate the enzyme once it's made by the cell. And then we generate a library that will contain, in this case, it was theoretical diversity of about 5 million different variants of UMOs. This is UPO. So this is 5 million different combinations of mutations on that starting backbone. And those combinations are guided by the engineering pipeline that Tiago just went through. Once we have that diverse library, we can screen it using our ultra high throughput screening platform. So this is based on microfluidic droplet screening. This was actually the subject of our earlier webinar. You should definitely check it out if you're interested. But what this tool, this platform allows us to do is we can screen something on the order of 1 million different enzyme variants in a day. And so this allows us to look through very diverse libraries and sort those enzyme libraries based on their relative activity. In this case, since we were interested in substrate specificity, we did multiple screens with different substrates to identify which enzyme variants were active against one or the other of our substrates. Once we know, or once we have collected the pools of enzymes that are active against these different substrates, we have huge volumes of data. At this point, we turn back to our data science department to help us sort through all of this data and make sense of it. So Tiago can tell us more about that.
Sure. So as you can see, even though we just reduced the search space by orders of magnitude, we are still dealing with a lot of variants. And we're happy about it because our high throughput screening platform is capable of handling massive amounts of variants. And we can actually experimentally validate all of them with this approach. So at Allozymes, the entire screening, the sorting, the sequencing workflows are designed and performed in-house. After screening all of these millions of variants and sorting the designed combinatorial libraries, we get pools containing enriched sets of strain variants showing improved activity towards our desired reaction. So these pools are then sequenced in-house, and the data is processed using our developed sequencing analysis pipelines. Our sequencing data is safely stored in our cloud, while our custom sequencing pipelines retrieve the maximum information available in these variants. So these sequencing pipelines are semi-automated, and they can be run by any scientists on our company immediately after the sequencing is started. So there is no waiting times here to send data elsewhere to be analyzed. They are used to highlight these sequencing pipelines are used to highlight high-performing variants as well as low-performing ones. So with this information, we can perform data analysis and identify specific variant sample enrichment factors, ranking individual variants for experimental validation. This means that we can select variants that have a much higher enrichment, meaning that they are much more active for the particular target reaction that we are searching for. With these obtained enrichment factors, we can also, for each individual variant, we can also create rich datasets that associate a large number of identified variants to individual fitness scores. As you can imagine, these are large data sets, and this data can be fed into different machine learning algorithms to train models to guide our enzyme-directed evolution. These models are used to predict unseen variant combinations with potential for higher activity. And with this approach, we can also explore other mutation paths that escape the local maxima and provide better predictions with different sets of combinations. What we're trying to do is not getting a local maxima that has just a specific set of mutations, where elsewhere might be another set of mutations that has even higher maxima. So these approaches can help us trying to escape that and navigate towards even better predictions. With this, Pradeep can again run experimental validations in the web lab.
So to summarize what we've done to this point, right? We somehow identify a good starting point that might be through our discovery pipeline, or maybe the customer already has a good starting enzyme. We then use our engineering pipeline to identify what diversity we think will confer beneficial changes to the phenotype. And then we use our screening to kind of cull that list of diversity down to the things that experimentally we see are improving the activity of the enzyme. So at the end of that, we get a generation of leads. So these would be, in this case, it was 20 combinations of mutations that we predicted would be beneficial based on our microfluidic sorting data. We then went back to our high fidelity direct product measurement assays. In this case, we measured 2 of these products by gas chromatography and 1 by, again, HPLC, so high performance liquid chromatography. And we were able to determine the activity profiles for 20 different lead UPO variants. You can see here in green, one of these enzymes, these combinations of mutations really bumped up the activity of the wild type enzyme against all of the substrates tested and to form all of the products. This enzyme in yellow, on the other hand, we see increased production of the desired product, which is that styrene oxide, but decreased production of the undesired products. So this is a more specific product profile, and this is what we were trying to target. So by using all of these tools put together, we were able to identify 2 enzymes that really had very different activity profiles and could be used for 2 very different customer applications. On the left, you'd have that green enzyme that was more active. In a lot of cases, we might not know what the customer's target substrate is, so maybe just having a more active enzyme might be the best one to start with. And in the other case, something that's more selective. So when the customer already knows what they want us to synthesize, we can target that. And so I think with that, we've not only met whatever the customer's needs are for their application, but we've also built up a large database of information about how mutations affect the activity profile of this enzyme class. And Tiago is going to tell you about how we can use that to accelerate future projects.
Just to recap, so by deploying both our discovery and engineering workflows together with this extensive experimental validation, we were able to find suitable enzymes and evolve them into high-performing or high-selective or both variants, which is great, and at the end of the day is our ultimate target. Nonetheless, this data from this ultra-high throughput screening of millions of variants and also the sequencing data that we harvest can be used to populate our internal database. Building upon these dozens of variant screenings, we can train or fine-tune foundational models that will ultimately help us accurately generating new variants or identify even new substrates for existing enzymes. That's it. So at Allozymes, right, we evolve enzymes. Whether you needed to develop a new enzyme, improve an existing one, or unlock new processes, our computational platform scales and your goals and will adapt to your chemistry. Thank you so much for listening, and we'd love to hear what challenge you are facing in enzyme discovery or optimization and explore how can we help you. Thank you.